All right, so we are now broadcasting, folks. So it has gone live, and welcome to everyone that is just coming in. We're going to kick things off in just a minute, but for those of you that are just joining, welcome to session 2.2, Advances in Rare Disease Therapies. We're really excited to get started, and we'll open with some brief housekeeping remarks once we get everyone in. All right, folks. So we are going to go ahead and get started now. And I know we still have a few more folks kind of filtering in, but just thank you all so much for coming back for day 2. And I hope that everyone who was able to attend the fireside chat this morning had a wonderful time. I think there was a great discussion there, and excited to kick off this next part of our session on advances in rare disease therapies. Before we get everything kicked off, I just wanted to review a few brief housekeeping notes. First, all attendees are currently muted by the moderator just to reduce the interruptions in the flow of the conversation. We encourage you to use the chat function in the Zoom window for any questions that you may have throughout the session, and the moderator team will present them to the speakers. To open the chat, please just use the chat button at the meeting controls at the bottom of your Zoom window near the mute icon. If there are any questions that are not able to be addressed by the speakers or we run short on time, we will do our best to follow up on those after the program, and we'll be taking note of any questions that we are not able to get to. If you have any technical difficulties or questions about the program as a whole, please visit the help desk in the main lobby, where you can chat with our tech team and the Global Gene staff members as well. Finally, we encourage you all to participate as active listeners. Take advantage of this opportunity to hear from leaders and experts here and ask questions of them. This program is your time to learn and find answers to your questions and critical challenges in rare disease research. Now, without any further ado, I'm so pleased to welcome all of our speakers here today. First, we have Tracy Dixon Salazar of the LGS Foundation and the Chan Zuckerberg Initiative, who will be our moderator. We also have Anita Hill of Alexion Pharmaceuticals, Catherine Beaverson and Pushkal of Pfizer, and Pushkal Gargavel-Nilam. They will each share a little bit more about their background during their conversations and presentations, but for now, I am very pleased to invite Tracy, our moderator, to kick things off.
Hi, it's so great to be here. It's great. I see you guys all filtering in. So I'm really excited about this panel. I think we're really excited to hear about the different types of therapies that are coming out. We're going to be talking about gene therapy. We're going to be talking about pharmaceuticals. We're going to be talking about RNAi. And we're going to be hearing from some leaders who really have embraced the patient voice, the advocate voice, and moving that forward. And hopefully we can give you some real examples of what is going on in the space that all of us want for our rare disease. So just to introduce myself really quickly, I am Tracy Dixon Salazar. I work at the Lennox-Gastaut Syndrome Foundation and also at CZI. I have a 27-year-old daughter with a rare disease, so I have been in the space for a very long time. She has something called Lennox-Gastaut Syndrome. She started having seizures, and that was really the primary feature of her disease. Seizures, progressive intellectual disability, and risk of early death. And that was our life for 16 years, just seizure after seizure, thousands and thousands of them. And back then, there was nothing really. There was an epilepsy community that was more focused on, not the rare epilepsies, because there's no market for those, but the common epilepsies, which is still important. There really wasn't even internet back then. I had to check it out at the library for an hour at a time. And so when I look at today and what we have, with Global Genes and with, pharma and FDA and others embracing the rare disease space, I'm so excited by it. What I did do during that 16 years of hell was I went back to school and got a PhD in neuroscience to try to help my child. I ultimately did find the cause of her epilepsy and was part of the genomic revolution for all the genes that we have today. I was one tiny piece of that, and I'm excited. But I moved into patient advocacy because the scientists had found all these genes, but nobody was really pushing them forward. Patients weren't getting tested. And it's just so exciting to see how far we've come. I feel like that older generation were like, back in my day, but no, it's really exciting to be a part of this. I'm so thrilled to be here, and I'm really excited, and hopefully can give you some really practical tips today from our panelists who are going to be speaking. So with that, I want to introduce I know that we just had an introduction on our panelists, but I'm just going to introduce them and then they'll go in order. Anita Hill is the Vice President and Senior Consultant of Hematology and, and the complement system at Alexion Pharmaceuticals. She's going to go first to be talking specifically about advances for rare disease therapies, telling the story of how patients really help to bring forward those pharmaceutical therapies and others. And then we have Catherine Beaverson, who's the Senior Director and Patient Advocacy Lead in the Rare Disease Unit at Pfizer. She's going to be talking about gene therapy and how patient advocacy really enabled those efforts at Pfizer. And then we're going to be talking to Pushkal Garg, who's the Chief Medical Officer at Alnylam. That's a mouthful for me, sorry, it's still early over here on the West Coast. And he is going to be talking about RNA therapeutics and specifically their global company and the advances that they've brought. So I am so excited. And Anita, would you kick it off, please?
Thank you very much indeed, Tracy. And it's wonderful to be here and talk at this conference about how we're advancing the care for patients with rare diseases. This is something I've worked on for approximately 20 years, firstly as a physician and now very recently in the pharmaceutical industry. So I hope I can give a perspective from both sides. By focusing on one particular disease, I'm going to walk through how a targeted therapy for a disease was developed, how it requires a whole team approach, and how pivotal the patients were in developing a world-class service, and where the company Alexion, who I now work for, are wishing to further the care of patients even more. To show how we all work together as patients and carers, physicians and the industry to make these advancements, I'm going to concentrate on a rare disease called PNH, which stands for paroxysmal nocturnal hemoglobinuria. And in order to develop a targeted therapy for a disease, one must first know how the disease develops. So how do we develop a disease like PNH? Well, first of all, we should note it is an acquired problem. This is not inherited. So in fact, it can happen to any one of us at any time. To explain it simply, if we look at our bone marrow, and we have these important cells in the bone marrow called the stem cells that produce all the blood cells that circulate in the circulation. So the stem cells can regenerate themselves, but they also produce the red cells that carry oxygen, the white cells that fight infection, and the platelets that we need for blood clotting. Now, randomly, to any of us at any time, one of our PNH stem cells can change, or what we call mutate. And this PNH stem cell, or this stem cell, can become what we now call a PNH stem cell. And actually, we could be sitting here now ourselves with a PNH stem cell in our bone marrow. These PNH stem cells also produce blood, but now we call them PNH red cells, PNH white cells, and PNH platelets. And I want to show why we call them this rather than the normal blood cells. And what I hope you can see from the cursor of my mouse is on the normal blood cells, I've put a thick black line. And this is to show on these normal blood cells that normal blood cells have an effective shield on the surface. It's a shield of proteins. But PNH blood cells, as you can see here, are naked. They don't have that shield of proteins on the surface. So these blood cells, importantly, can be attacked by parts of our immune system. Now, why then do we all not develop this rare condition? Well, that's because you need something else other than just a PNH stem cell. And that's where your body, a part of your body, the immune system, attacks the normal blood cells and reduces them. And you can see if you have lower numbers of normal blood cells, that your red cells, your white cells, and your platelet count will fall. Despite decades of research, we still don't know why this immune attack on the normal stem cells doesn't affect the PNH stem cell. And therefore, these PNH stem cells can now be considered as being stronger, really survival of the fittest, and then produce more of themselves and produce more PNH blood cells in the circulation. So all of these PNH blood cells can now be attacked by part of our immune system. And that part of the immune system is called the complement system. So think of complement that we all have as a series of proteins or bullets designed to destroy foreign organisms like bacteria or viruses. We should have this shield of proteins, CD59 is one of those, on the surface of our cells to try and protect us from this complement attack. Now you can therefore see where therapies might work, because PNH blood cells have lost that shield of proteins and are therefore easily attacked by this complement immune system. So therapies to protect PNH blood cells have been in development. And this was the first drug I was involved with many years ago when I was a clinician, where we looked at trying to protect PNH blood cells. But this compound ceased development, and the drug eculizumab was developed. And what this drug eculizumab does is bind to the middle of our complement system. It's called a complement inhibitor. And by binding to the middle of the complement system, you prevent this end complement activation, and it leaves our blood cells unharmed. So this shows how knowing how a disease happens is how we can develop a therapy. So we had got this far, but we now needed to know how to develop it further to give to patients. So this is how we developed administration of this therapy to patients in the UK. And although this is an old slide, the principles are the same in that the Department of Health funds our public National Health Service. And what I want you to focus on is this group here, the £500 million approximately that are given to a group called the National Commissioning Service. Actually, they keep changing their name, but what this NCG equivalent group does is fund patients with rare diseases. And this is incredibly important, because if we didn't have a national service for rare diseases, there could be unfairness around the country on who gets treatment. And it could be what we want to avoid, be postcode driven. So we want this national service, which has to be highly specialised. And this is where the patients came in for us, because a minister has to recommend that a disease is highly specialised in order to get national funding. And we as the physicians all got turned down when we applied for a designated status for this rare disease. When the patients found out, they wrote to their own members of parliament. A big group of patients did this. And that meant that one in particular got very heavily involved, wrote to their MP, who was then pivotal in us developing this world-class national PNH service, as well as provide an invitation for us all to go and visit the Houses of Parliament. So it really was down to the patients that we were able to develop this national PNH service. We therefore gave the drug to children, elderly, and everybody in between, and we significantly improved survival for patients with this rare disease. So we've already improved survival, but Alexion have decided not to stop there. They have a commitment to continuously advance the care for patients wherever possible, to continue to innovate and provide even more options. So they do this, for example, by looking at the frequency of administration and also looking at other options. And in this final slide, you can see the drug eculizumab is what I've used for 18 years and was actually approved after the clinical trials in 2007, and it improved survival. But Alexion decided that for further quality of life of the patient, having an 8-weekly infusion means a patient can forget about their disease, hopefully, for approximately 2 months. For even further development, the concentration was improved so that the administration could be even quicker, again improving a patient's quality of life. Even further, to provide options for the patient, a subcutaneous route has been delivered, which for some patients would give more independence. And an aspiration for the company is the development of an oral tablet, which you could also see would have a huge impact. Again, patients have a huge role to play in the global PNH registry. We have huge learnings from the registry, and this is an enormous way that patients contribute to future developments. So I hope that does show that through the partnership between all of us, the industry, the physicians, the scientists, and the patients and organizations for those patients, allows us not to become complacent and always strive to improve the care for patients with rare diseases. Thank you.
Thank you so much, Anita. That was fantastic. I love how you emphasize, the advocacy, the involvement in the registry and the trials, and how we really all need each other. That's fantastic. Catherine, you are up next. Why don't you take it from here and tell us about gene therapy? Catherine, you're on mute still.
Apologies for that. Thank you very much for the introduction, and thank you to the organizers for inviting me to present, and thank you to the folks who registered for this session. I'm really pleased to be here today. As Tracy said earlier, I lead patient advocacy for Pfizer's Rare Disease Research Unit, which is the advancing.
Catherine, you froze for just a second. So if you want to go back, I'm not sure if you froze for everybody, but you froze for me. So if you want to just go back a little bit, that would be fantastic.
Go back with?
You cut off for me right after you started to introduce yourself. So I'm not sure how far you got.
Oh, okay. So, right, I lead patient advocacy for Pfizer's Rare Disease Research Unit, which is responsible for discovery, translational sciences, and early clinical development. So I have the unique opportunity to work with patients kind of early on in the drug development process. And my screen is frozen. I can't advance. Okay. Yes, in fact, I am an employee of Pfizer. All right. So at Pfizer, we're very excited about the potential for gene therapy to address the significant unmet needs of individuals and their families burdened by rare diseases in order to transform their health and well-being via this one-time treatment. Our current focus is on the development of investigational gene therapies for hemophilia, both A and B, Duchenne muscular dystrophy, and rare diseases involving the CNS and inborn errors of metabolism, inclusive of Wilson's disease and a genetic form of ALS. And in this session, I was asked to give a brief introduction to gene therapy, describe some of the scientific challenges in drug development for gene therapy, and to provide some examples of how we've done the bulk of our work in this program with patient organization and patient communities to advance the program. And I'm going to focus on a lot of those examples in the work that we've done with the Duchenne community. So what is gene therapy? It is a therapeutic strategy used to replace or repair a dysfunctional gene. It's most applicable to diseases caused by a single gene mutation or monogenetic diseases. And it is an approach that aims to improve symptoms or cure a disease or correct a disease towards health. The workhorse here is the vector, right, which is the delivery system of that therapeutic gene to a particular target. And the right-hand side of this graphic really focuses on the fact that vectors work by traveling to the target cell, right, binding to the cell and entering. The therapeutic gene is inserted in the cell, whereby the cell's machinery kind of takes over to produce a functional protein. It's important for this group to understand that there are multiple types of vectors. Pfizer is focused on advancing adeno-associated viral vectors, which has a capacity limited to 4.7 kilobases. And when we think about the scientific challenges of AAV, I'll kind of point to, the challenges in Duchenne specifically, in that there are multiple considerations in vector design and packaging, right? Can you get the right DNA into the vector? The dystrophin gene is too large for the AAV vector, so that current investigational approaches are using gene therapy or truncated versions of the dystrophin gene. Can you get the vector to the target in an amount that is enough to constitute a therapeutic dose? For Duchenne, we've had to think about the AAV serotypes and vector designs that target skeletal and cardiac muscle. And we all know the muscular system is one of the largest organ systems, so one might anticipate a need for high doses of vector. Do we understand the immune system? And Anita did a great job of teeing up, the immune system. How will it respond to the vector we're using, or even the mini dystrophin gene that we're introducing? And what's the best way to address those immunological challenges? And then do we understand what level of expression of the corrected gene is needed to improve function? And ongoing clinical trials for Duchenne are attempting to learn and define the level of the miniaturized dystrophin expression that's required to improve muscle function. So I'm not going to go into detail on this slide, but I think it's important to realize that the gene therapy programs we see today are a result of the pioneering vector biology work, translational research that, Christopher Austin, so nicely teed up yesterday, and the early clinical research work spanning more than 30 years by leaders like Jim Wilson and Jude Samulski and Professor Zhao Zhao and Jerry Mendel. And much of this work was enabled through grant funding from patient organizations like the MDA and Cure Duchenne, such that when Pfizer acquired Bamboo Therapeutics in 2016, we were able to move this program forward and into the clinic with the first participant dosed in early 2018. So when we, where are we now in these programs? We have an ongoing phase 1b study, right, of a mini dystrophin gene therapy, with the primary endpoints being safety and tolerability. We anticipate starting a larger global study later this year, which is looking at the primary outcome being efficacy as determined by change from baseline in the North Star Ambulatory Assessment over 1 year. Okay, so I'm preaching to the choir when I say that patient organizations really possess the capabilities and expertise to lead, drive, or enable advances across the drug development landscape through, all these elements that are listed here, from early research optimization to policy advancement. And so the rest of my presentation is really going to focus on ways that Pfizer has engaged with advocacy to move some of these things along through collaboration to benefit not only our program, but the scientific community at large in the Duchenne space. And I'm not going to present a comprehensive list, but certainly examples that can get our audience here to think about where do we want our groups to build capabilities that's both possible within the framework of your organization, but then valuable in the landscape of drug development. So I think, most of the groups are familiar that, you can have a profound impact on a clinical development strategy, program, and protocol. And in fact, the Duchenne community have been wonderful partners in helping us with our protocol design, our recruitment strategy. When we think about diseases like Duchenne that are slowly progressing and we need lots of time to look at change over time. What is the burden placed on these families that we can, address through, addressing some of these retention issues? And then, how does the community work with us to identify and fill the gaps in education and communication needs? And, this slide highlights some of the activities that we've engaged in with the community. I think, in these early days of gene therapy, it's really important to tie in with educating regulators, right? They have expressed an interest in engaging with patient communities throughout the medicine's development process and to incorporate the patient perspective into their regulatory decision making. And so this example and the next that I'm going to show are examples of advocacy-led initiatives to generate evidence documenting the burden of disease and the community's goals for treatment. This Duchenne gene transfer study, Pfizer collaborated with multiple other entities. It was led by Parent Project Muscular Dystrophy, and the goal was to explore preferences and risk tolerance for emerging gene therapies using a snapshot of what we knew at the time that we started in this project about gene therapy. So through qualitative interviews, patients were asked to share their attitudes about and preferences for gene therapy as a potential therapeutic option. And then those identified attributes were measured through stated preference methodologies, a best-worst scaling experiment to measure priorities when deciding on participation in an early phase gene therapy trial, and then a threshold experiment to measure what's the maximum acceptable risk of death for a non-curative gene therapy. And all of this work is publicly available through publications. And then, as part of our involvement in the Jet Foundation's BioCouncil, we collaborated with them on a burden of illness study consistent with the FDA's patient-focused drug development framework to systematically collect burden of illness and burden of current treatments, and then to define the goals for treatment regardless of modality. And I think this study was particularly interesting because it defined treatment goals by assessing the things that mattered the most to patients: daily functioning, relationships, sports, and recreation. And the reasons they are important. Is it to maintain independence, self-esteem, enjoyment, in order to then determine the desired outcome for a therapy, right? Is it to improve, maintain muscle function? Is it to increase definition or independence, increase quality of life? What is it? So that there's a consistent voice of the community when speaking with regulators and each other. And then the last project I wanted to highlight is very important as well, because payers need to be educated on what matters most to patients and what the economic burden of the disease is in order to pave a path for access to these therapies. The Duchenne community has learned that approval of therapies does not ensure access and reimbursement. Pfizer has joined a multi-stakeholder collaboration led by Duchenne UK called Project Hercules, and the goal here is to generate a pharmacoeconomic model that all companies can use to provide the evidence that health technology assessment bodies need to make transparent and consistent reimbursement decisions. And so my final slide, and in summary, is that there are multiple and many opportunities to engage with industry to enable and advance drug development. In my presentation, I've highlighted a handful of examples where Pfizer has done this to advance our Duchenne program, as well as advance drug development for the community. I happen to be a big fan of the Clinical Trial Transformation Initiative model for patient group engagement across the clinical trial continuum, which is shown here. It's a terrific roadmap for patient groups and companies and academics to align their capabilities and their priorities with their strategy for participation in the drug development process. And, you know, I kind of leave that with you as a model, as your groups think to build their capabilities and expand in this domain. So thank you very much.
Thank you so much, Catherine. The muscular dystrophy community has just been a great model, I think, for many of us. So I loved all those examples. We're going to move right on to you, Pushkal. Why don't you take it away with RNAi?
That's great. And let me just get this going here. Well, wonderful. Well, first of all, thanks to the organizers of Global Genes. Thank you, Tracy, for moderating the session, and to the fellow presenters, and most importantly to the attendees. It's really a privilege to be amongst all of you who are, you know, connecting and advocating and empowering the rare disease community. So, my name is Pushkal Garg. I'm the Chief Medical Officer at Alnylam Pharmaceuticals, and we are a company based out of Cambridge, Massachusetts. That's been around for about 18 years. And we are working to turn a novel Nobel Prize winning scientific concept, RNA interference, into a therapeutic modality that can be used to address a number of genetically driven diseases. And we have now recently filed for our third drug approval, so we've got a robust product portfolio, and we are a global company. So let me take a couple of minutes, because you may not all be familiar with RNA interference. It's also known as RNAi, sometimes it's called gene silencing, and explain what exactly it is and why it offers a unique opportunity to potentially address a number of diseases, and particularly a number of diseases of concern to the rare disease community. So many of you may be familiar with what's called the central dogma of biology. DNA, which is our genetic code, is transcribed into messenger RNA, and then that is translated into into proteins, some of which can be disease causing. Right? It's kind of like having a blueprint for a house, and then you can have a detailed schematic for a particular part of your house, you know, a banister or something, and then on the far right is the actual banister. Now, we have a number of therapies that are available. Most commonly, we develop therapies, small molecules, antibodies that address the protein that may be causing a disease, the disease-causing protein itself. That could be an antibiotic that's targeted against a bacterial protein, or a native protein that may be causing a disease. On the other extreme, we have therapeutics that are coming that can address the DNA. And Catherine's spoken very eloquently about gene therapy, for instance, as a way to address the DNA. And what we do at Alnylam, and some other companies are working in similar areas, is work on the middle portion. We actually develop small silencing RNAs that can actually stop and focus on the specific blueprint for a particular protein and silence that protein. And so we have a focused way to dampen down the production of one particular specific disease-causing protein. So we've been working on that technology, and this is what it is. We basically can introduce something called a small interfering RNA. It's typically given either intravenously and now most commonly subcutaneously. It travels to the site of where that disease-causing protein is being made, and it chops up that messenger RNA so that the disease-causing protein can no longer be made. All right? So that's what we've been working on. And through that technology, we now have a portfolio of medicines directed at a whole host of diseases. The first molecule that was approved was for a drug called Onpattro, which is being developed or has been developed for hereditary ATTR amyloidosis. I'm going to speak about that in a little bit of detail just to show you how we've approached drug development and how patients have helped inform that development program. Late last year, we had a drug approved for another rare disease called acute hepatic porphyria. That's called Givlaari. And recently, we filed for our drug called lumasiran, which is for a rare disease called primary hyperoxaluria type 1, which affects particularly very young children. And then you can see we have a number of medicines coming behind that, where we're trying to employ this technology to address unmet needs for patients with both rare and common diseases. Now, here's an example of how we've developed a medicine to address this very rare, progressively debilitating, and often fatal disease known as ATTR amyloidosis. Now, it's caused by a misfolded protein, transthyretin, and then transthyretin protein is produced in the liver. But if there's a mutation or a genetic change in that protein, it can accumulate as amyloid deposits, protein deposits that go into the heart, the nerves, and the GI tract. And about 50,000 people around the world have this genetic form of the disease that transmits through families or passes along through families. And then we've increasingly learned that there's a large number of patients who can actually have misfolding of the normal protein as well and have very similar disease manifestations. So we did so what does Onpattro do, which is the medicine that we developed? We developed this medicine. It actually binds to that transcript or the messenger RNA for the TTR protein in the liver. It's given through an infusion, goes to the liver, and there it can cause a reduction in the overproduction or the production of this altered protein. And as such, it lowers the protein levels. We found in a study it lowers the protein levels by about 84%. And then that results in fewer deposits of that amyloid protein in the nerves in particular, and possibly in the heart as well. And we're exploring that further. So how did that manifest? This shows you some of the primary endpoint data. We used a scale called the mNIS+7 for patients with the neuropathic or the neuropathy aspects of the disease. And in that neuropathy aspects, patients often have progressive trouble feeling things with their sensory nerves. They have trouble moving because of the effects and damage to their motor nerves. And then they also have trouble with autonomic symptoms. So they can get dizzy when they stand up. They can have alterations in their heart rate, etc. They can faint. And so we measured using this mNIS+7 measure all the aspects of their nerve function. And what we found is that patients who got the placebo therapy for 18 months had a worsening in nerve function by about 28 points. And what's interesting is that with Onpattro, not only were we able to slow the progression of that disease, but we actually saw that in about 56% of patients, their nerve function improved over the course of the study, suggesting that by really working at the core aspect of the disease, that you can actually prevent and maybe even remove some of the damage caused by this disease. So this was very encouraging. We also, and this is again where the patient voice was very important, we incorporated not only measures of nerve function, which are very important, but we also looked at the impact on patients' quality of life. How could they do and how did they feel over the course of the study and after getting the therapy? And we found similar to the nerve function that, in patients who unfortunately were not getting active treatment, they actually had worsening their disease, but the treatment actually improved their quality of life over the course of 18 months. The patients that we spoke to when we were designing the study told us a lot about the symptoms that they had. They talked to us about impact on their GI tract, diarrhea, constipation, dizziness, nausea, and vomiting. So we measured those aspects and found improvements there. Patients told us that they lost weight with this disease, and so we incorporated a measure to look at weight and found that there was actually a reduction in weight loss and even weight gain in patients who were treated with active therapy. They could ambulate better, crossing the street faster. They had less pain, and importantly, it improved symptoms such as anxiety and depression. All medicines have side effects. Patisiran is given by an infusion, and some patients can get infusion-related reactions. This can typically be managed by slowing down the infusion rate or giving certain pre-medications right before the infusion. And it can affect vitamin A levels. And so patients who take this drug need to also take vitamin A supplementation. I spoke to you a bit, and we can talk a little bit more in the question and answer period about how we've engaged the patient community to inform the design of our clinical trials, but that's just one portion of it. You know, we used patients to highlight the unmet need. We use them to help us understand what are important clinical trial endpoints. We use and work with the patient community and groups like the Amyloid Support Group and other groups in terms of furthering patient education efforts. We've developed a program called Alnylam Act, where we're able to provide free genetic testing so that patients can get a genetic test if they need it to see if they may have this disease and get earlier therapy. Importantly, it's critical that, as Anita highlighted, that when we develop these therapies, that we're able to provide access so that as many patients as possible can get these new medicines. And so we've partnered and entered into innovative programs with payers and insurers to try and widen the net as much as possible of patients who can avail themselves of these therapies, funding foundations. And we have a very vital and active patient support system called Alnylam Assist, because the healthcare system is so difficult to navigate at times, so that patients can find out how to get assistance, get directed to an appropriate therapeutic provider, get the support that they need, and all get assistance on how to navigate the funding and reimbursement for these medicines. So that's a very active part of how we think we choose to engage the patient community. So as I've tried to highlight, you know, Alnylam's approach, and I think all of our approaches from the people you've heard on this podium today, is to really engage with patient advocacy group and the patient community through the entire life cycle of drug development and into when these medicines become available for broader use, to increase disease awareness, speed time to diagnosis, gain insights into our new programs that we may be working on, supporting clinical trial enrollment, engaging policymakers, and enabling access to treatment. And so it's a very exciting time, I think, broadly in the biotech and biopharmaceutical community because of all the new technologies that are coming forward. We're proud to be part of that. And again, we believe it's really critical that patients and patient advocates, like the people here, are actively engaged in that process. So I will stop there and turn it back to Tracy for any question and answer.
Terrific. Thank you guys so much for your comments. I think this is really exciting. You guys are all taking this very holistic patient approach where patients are co-developers and co-designers with you across the spectrum, right? And you're not, you know, pigeonholing them into one area where they don't have input, because only patients can speak to the burden of the disease, to the risks they're willing to take, right? All of these things. So I have not necessarily with our rare disease had that, and I think maybe some of the people on this webinar have had that sort of holistic approach to patient advocacy input at therapy companies, you know, and so maybe I'm doing it wrong. Maybe I need to go work with Alnylam and Alexion and Pfizer more. But how is it that, you know, if we are interested in science, we are pushing it forward in our rare diseases, you know, what is it that convinced you, I want to ask each of you, to you know, focus on RNAi in your disease, in that disease state push call, or, you know, to work with one group over another, or can you work with all of them, Catherine? Or for you, Anita, in the European system, how did you decide? Because if there's 7,000 or more rare diseases and 7,000 plus organizations, you know, how is it that we can move forward with our disease? What is it that we need to be doing as young advocates and that maybe aren't there yet? Pushkal, do you want to go first?
Yeah, I mean, I mean, you threw a lot of questions, and I think, you know, it's look, I think what I would say is one of the things I'm proudest of, I've been in the industry for 20 years. I think, you know, as you highlighted, there's really a sea change in how we think about incorporating the patient voice, right? And I think to your point, that wasn't always the case. Healthcare hasn't always incorporated the patient voice. And certainly drug development hasn't always incorporated the patient voice. But this organization and many others, I think, have really brought the patient voice to the forefront. You know, Catherine highlighted that we have groups like the CTTI that have kind of mapped out a roadmap. There's patient-focused drug development and patient voice meetings that the FDA sponsors. So for example, for ATTR amyloidosis, for some of the other programs for porphyria, those patient voice meetings were incredibly influential. I can't overestimate how critical it was for drug developers and for regulators to really hear the patient perspective. What are patients going through? Many of these diseases that we're talking about, most physicians, most regulators have never heard about. They've certainly not seen a patient with these diseases, right? And there's nothing like seeing and hearing from the voice of a patient or a caregiver or an advocate what the patient has experienced. You know, what is it like? What is the burden of that disease? How does it affect their day-to-day life? And it shapes, you know, so many aspects. First, it promotes people looking into those diseases because they recognize that there's an unmet need. We have so many more therapeutic things in our armamentarium, whether it's antibodies or small molecules or RNAi or messenger RNA or gene therapy or CRISPR-Cas9, all these different things that we can now use to attack these diseases, think about the right endpoints, and importantly for regulators, what does it mean in terms of benefit risk? What are patients willing to accept in terms of enrolling in a trial and potential side effects for disease that they may have that they're suffering from? And then ultimately for payers and physicians in the community to understand what patients are going through and to make those drugs available to patients in their health plans or in other settings, right? So I think there's places to attack every step along that cascade where patients can have influence. I'll turn it over to others who might have other things to add to that.
Now, this is Catherine. Tracy, I'd love to address your question around, you know, how do you choose whom to work with, and you know, what are you looking for in those engagements? And I think, you know, we all know that there are many groups in Duchenne muscular dystrophy who are passionate and engaged, and I think, you know, organizations such as Pfizer and the advocacy organizations, you know, kind of need to look at their strategies, right, and their capabilities, and are they aligned, you know, over the right issues and the right timing, right? And for those organizations that have built their capabilities in around the science, right, who has the mouse models, or who is, you know, funding early research, right? There's maybe a time for strategic engagement around those capabilities. And then groups who are more focused on policy advancement and engaging, you know, with legislators or payers, right? You know, then you partner around, you know, with those groups for that initiative. But I think what's important is that there's transparency around why you're working with groups over what activity, right? Because, you know, we want to be working with, we want to feel inclusive and be inclusive of the entire community. But you understand that, you know, there might be a strategy in place for earlier engagement versus, you know, later engagement. And that's one of the points that I got to around, you know, kind of engaging with the community early on to understand what are their needs for communication and education, because ensuring that they will be brought in, right, is an important message.
Anita, tell us about the UK system. How do you decide which, I know it's a totally different system across the pond.
So it's a really interesting question, because the example I gave showed in a disease like PNH, where prior to eculizumab treatment, we had no treatment for this disease. So in fact, there was no patient organization, because often that comes when, you know, you've got more than supportive measures, which is what we had. When we were turned down twice to develop the National PNH Service, a group of patients came together. Now that ended up evolving into a patient group, and I should have called out, and I'm glad I've got the opportunity to do it now, a very active patient support organisation called PNH Support, and also PNH Scotland, so we cover the whole of the UK, and they have remained very active and ensuring education and materials for patients, and being involved in developments now. So even though we've got a very effective therapy, we know there's more that we can be doing as well. And so they're saying to us, are you looking at quality of life in your clinical trials? They're questioning us as we go. And that was when I was in the medical profession, and now in the industry, we also incorporate them in listening when developing trial designs. It's having that voice. So I think what I've had the advantage of seeing is from having a disease with no treatment to then having a patient organization develop not just one treatment, but further treatments in the future.
I love that. Yes, Francis Collins also said that he would not want to have a rare disease that didn't have an advocacy organization associated with it, right? And there is an element of sadness to that statement, but also truth of the way that the system works. So I just want to say to our audience, please take a moment to type in your questions. We have about 10 more minutes total for questions. And so we would definitely want to get those answered. You know, I want to ask, you know, specifically, you know, if you can, just to dive a little bit further down. I know I've already sort of answered this question, but what is it that I mean, I know you're looking at your strategy, and I know that you're looking at, you know, where the patient organization is, but what is the linchpin? Is the linchpin, I'm going to work with this patient organization because we already developed drugs in this area? Am I going to work with this patient organization because we already, you know understand that gene? Am I going to work with this patient organization because I met this mom on a bus and I felt bad for, you know, for what she's going through? All of the above, right? And can you guys give us any insight? Because a lot of us are really trying to get pharma to even have a conversation with us, right? Let alone, you know, really go all out the way you guys have. So any insight? I know I'm pushing this question a little bit further, but it's okay if you don't have an answer, but I'd love to know.
Yeah, I mean, maybe Tracy, I can start. I mean, I think kind of what you're asking is a little bit of, you know, how do we decide which diseases to go after? I think is really at the core of what you're asking. I mean, so, you know, when we focus at Alnylam, we're focused on RNAi therapeutics, as I mentioned. And where our therapeutics are best suited is when there is a particular excess of a disease-causing protein. Because what we fundamentally do is we turn off the spigot on protein production, right? We pick a particular protein and we turn off the faucet so that that can't be made anymore. And so what we have is, you know, a group of physicians and our researchers who are working together, and our scientists working with physicians around the world to really identify diseases where there's one or two more disease-causing proteins that are elevated, where we know that we can hopefully safely bring them down and affect the trajectory of a disease. And so some of the diseases I highlighted, TTR amyloidosis is caused by production of this abnormal TTR protein or overproduction in certain settings, right? Porphyria, we know hepatic porphyria is caused by a regulator protein called ALAS1, and we know that we can bring that down. And so there's a number of diseases like that. And so we come at it from a biology perspective because we're developing a new therapy and we need to figure out where are the best chances for success from a scientific perspective that we might be able to have an impact on disease. But that's just the beginning part of it. We have to marry that very, very early on with understanding what is the magnitude of the unmet need. Would we be able to actually demonstrate that we have a favorable impact on patients' health? And so soon after we engage in that initial scientific discovery effort, but well before we've entered into a clinical trial, right, when we just have a concept, we start to look into, and we have a very active patient advocacy group. Who are the patient advocates out there? What are the groups that are focused on these particular diseases? Do they exist, right? We talk to clinicians who specialize in treating those diseases, and then we identify patient groups that may be focused so we can understand the burden of illness and the experiences of patients with that disease. And we bring those two things together, unmet need and scientific tractability. Is that a disease that we think we can hit with our therapy? And those are the two things that allow us to go forward. And then we really try and focus on maintaining those relationships with those advocacy groups and patient groups as we go further, because as we've highlighted, there's opportunities all along to influence the drug development path. I hope that helps.
That does. So I think just repeating back, as a small foundation that's getting started, as they go to build their capacity, it's really important for them to understand the biology of the disease, at least what's known about it, right? And say, listen, Alnylam's not a good option because we don't need the spigot shut off for our disease. We need something completely different.
That's
And
exactly
they can
right.
work with their scientists and clinicians to understand that. But then they also need to understand from their patient community what the unmet need is, right? And maybe not just their own anecdote, but also the broader community of, how common are these symptoms and what are the symptoms, and then how would patients prioritize that, right? So as they're building capacity, it sounds like those are two, there's a lot of questions that have to be answered, but those are two
Absolutely.
hugely important for drug development.
And engaging with physicians. Oh, sorry, Kath, just one last point. And engaging with physicians who are focused on treating that disease, right? Because that sort of becomes a great ecosystem where those discussions can happen. Sorry, Catherine.
No. I was just going to emphasize a point that you made, Pushkal, around the science, right? And I think, understanding the scientific tools that drug developers need to understand not just the patient community, right? But the disease itself. And are these tools available for industry, to use? We can't, natural history studies, right? Do we have a good understanding of how these diseases progress, and how can, advocacy be enabling natural history studies, registries, the development of endpoints, really knowing the biology and ensuring that it's in the public domain, that the academic researchers that are being funded are publishing this for use by the broad stakeholder community. And the advocacy is, advocacy groups are that pivot point to all of the stakeholders. And do you really understand the gaps in data that are needed to move a drug development forward, and how are we filling those gaps together?
Great. Well, we have time for one last question, and I want to relate it again back to our advocacy groups. We're really trying to build up this development. A lot of times there's more than one advocacy group for a disorder or disease. And so how do you guys work with, if there's more than one for ATTR amyloidosis or Duchenne or PNH? How do you guys work with those? And, are there any tricks or tips that you can provide to or advocacy organizations that are building their drug development pipeline pieces? Anita, did you want to go first? We haven't heard from you in a few minutes. It's okay if you don't.
No, it's fine. I think for a disease as rare as PNH, and obviously with the company Alexion, there are other rare diseases they're also involved with. We tend to go very country specific, but there's also what we call the Global PNH Alliance that will then look at many countries together, particularly in Europe. And I think this is where a lot of the messaging, the same thing that Pushkal said, where we have gone to these groups is where we want to understand the unmet need. And that's such an important part. And this is why even though we had one very effective therapy that significantly improved survival, why companies don't stop there. Because we're looking and we're hearing, well, actually, we know the drug works really well, but do we really want IV every 2 weeks? Could we have something else available? So we might go to different countries and organizations depending on reimbursement and the groups that already exist.
Fantastic.
The only thing I would just add, I agree with everything that Anita said. And I think, we don't consider ourselves, we're not looking to sort of pick one advocacy group over another. Really our goal is to engage the global patient community for the disease. We develop our drugs and medicines, and we're motivated to do that, to treat, full swath of people who are affected by these things. That's our mission. And so we do look to engage all the groups that we can. In fact, our advocacy group even created at one point, last year, a summit where they brought together a number of the advocacy groups to really see, where were the connections and foster some of that. So that was an opportunity we took to try and bring the global advocacy community together within diseases, but even across a couple of the diseases we're working on, to get feedback on what we can do better, but also to enable perhaps more younger advocacy groups. Certain diseases didn't have the same level of experience in advocacy as other disease groups do. And so how do we sort of facilitate that mentoring and shared best practices? And so that was something that our advocacy organization championed last year and was able to off, which was very successful.
Very nice. Well, we do have a couple of questions that we didn't get to, and we will definitely respond to those. We're noting those. I want to thank each of our speakers for coming here and taking the time to tell us the amazing their work that they're doing to engage patient advocates across the drug pipeline and what we're all aspiring to, and also to talk about the different modalities, which I think give all of us in the rare space a lot of hope, right? If it's not RNAi, maybe it's going to be protein replacement. If it's not protein replacement, maybe it's going to be gene therapy, maybe it's going to be a biologic. So we have a lot of options out there to pursue. So I think that was very inspiring to hear these are moving forward, and that like you said, Pushkal, we didn't always have interest, you know, in the patient voice and patient input, and that is so shifted in this day and age. We're grateful. I hope to you guys that were listening that this was helpful to you. I know many of you are young and on this track to get your groups up and started, and I know that any of the speakers would be happy to follow up with you and talk with you if you have specific questions. And with that, I just want to say thank you so much. Thank you, Global Genes, for allowing us this platform to chat, and I hope you guys have a great day. Thank you.
All right, thank you.
Everyone so much.